So I guess you know what I'm thinking about.
Good morning, everyone. It's a great pleasure for us to host Dr. Orest Blaschuk, the founder and president of Zonula Incorporated, with us today. I apologize for the technical issue that we had, which delayed the start of this presentation for a few minutes. But for those of you who have been with us since during the past 2 years, we have had these issues before as well. And then I would like to thank you for your patience. So, without further delays, I'd like to introduce Dr. Orest Blaschuk, who is the founder of Zonula, a privately held Canadian biotech company developing therapeutics targeting cell adhesion molecules. They call them biological glues. And these molecules are called cadherins. He has worked in the field of cell adhesion for over 3 decades and was a tenured associate professor at the Faculty of Medicine at McGill University for more than 35 years. He was retired in 2021. Before starting Zonula, Dr. Blaschuk co-founded Adherex Incorporated in 1996, which is a publicly traded McGill spin-off oncology company, and where he served as a chief scientist between 1996 and 2005. Adherex developed anti-cancer drugs targeting cell adhesion molecules based on intellectual property generated by Dr. Blaschuk's laboratory. The Adherex drugs, ADH1 targeting the cell adhesion molecule and cadherin, was given orphan drug status by the FDA for the treatment of melanoma. Dr. Blaschuk has proven expertise in drug discovery and development from laboratory to the clinic. Dr. Blaschuk and his colleagues at Zonula have recently discovered a new generation of cadherin inhibitors, and they're developing these for the treatment of fibrotic diseases. So in this talk, he will discuss the history of cadherin inhibitors and their potential therapeutic applications. Thank you very much, Dr. Blaschuk, for accepting our invitation. It's a great pleasure for us to have you here with us. Before we start, I just wanted to mention to the participants that if you have any questions, please use the Ask a Question box so we can relay your questions to our speaker. So the virtual floor is now yours.
Thank you. Thank you for inviting me. I've had glancing meetings with several of you, and it's nice to be able to give a presentation on my work. As you can tell by my first slide, I'm Ukrainian, and the events have been very troubling to me personally, having been born in a small Ukrainian town called Winnipeg. If anybody wants to contact me, my email is given on this slide, and also you might have a look at the website for the company Zonula. There's a lot of information, and the news page has a lot of stuff on it. Next slide, please. So the presentation, I'll give some introductory remarks, which I've already started, obviously. Talk a bit about the structure and function of cadherin, and then talk a little bit about antagonists and agonists of this adhesion molecule. And then talk about some fibrotic diseases, which we're investigating using our antagonists. Next slide, please. And I just had a review article published on this topic. So if you're interested or in everything I'm going to say today is in this review article. Okay, so it's there. Have a look. Next slide, please. What I've been working on for, it seems like all my life, is what I call cell adhesion technology. Cell adhesion obviously is a fundamental biological property. Multicellular organisms wouldn't exist without cell adhesion. If you could manipulate adhesion, in other words, if you can manipulate how cells stick to each other, you could manipulate a wide variety of processes, and hopefully use that technology to treat a variety of disease states. My technology is focused on a group of cell adhesion molecules. They're called cadherins. You could think of cell adhesion molecules as glues because they glue cells together. There are a lot of cadherins. There are 114 human cadherins, so there's a lot of work to do. And I'm focused on only one cadherin, N-cadherin. So there's plenty of room for anybody else that wants to work on cell adhesion. Obviously, cadherin antagonists and agonists can be used to treat a variety of diseases. An antagonist is an inhibitor, and you could also think of it as a solvent. And an agonist is a stimulator of a process, and in this case, you could think of it as a glue. So just so we're clear on what an antagonist and agonist was. Next slide, please. Great. Yeah, next slide. So this is the target. This is the beast that I've been trying to trap. It's a integral membrane glycoprotein. It's a single pass glycoprotein, goes through the plasma membrane just once. Is there? Can you see my indicator, my arrow?
No.
There's no arrow. Okay.
I can show.
Okay. Well, it's not. I'll describe it. So there's 5 extracellular domains, EC1 to EC5, transmembrane domain, and 2 cytoplasmic domains, CP1 and CP2. The cytoplasmic domains interact with a protein called beta-catenin, and beta-catenin sticks to another protein called alpha-catenin, and they then anchor the N-cadherin to the microfilaments of the cytoskeleton. On the outside, in the extracellular domains, in the first extracellular domain, that's the business end of the molecule. There are several key amino acids. There's a tryptophan, a W, standing for tryptophan, right at the end terminus of the EC1 domain. And then there's a site, histidine, alanine, valine, HAV, towards the end of the EC1 domain. And that's the business. These two, especially alanine, so the tryptophan and the alanine, are involved in direct adhesion. And it's also important to note that the phosphorylation of the cadherin or the catenins results in weakened adhesion. So in the case of cancer, adhesion is very weak because the kinases in the cancer cells are activated and they're phosphorylating all sorts of proteins. So adhesion is weak in cancer cells and also in cancer blood vessels. Next slide. Yeah, so beta-catenin, just a word about that. It has several functions. One is it binds to N-cadherin, but the other function is it goes to the nucleus and it acts as a transcription factor. So if you can believe that, it exists at the cell surface, it exists in the cytoplasm, and it exists in the nucleus. It's also a component of the Wnt signaling pathway, which is a very important pathway both in cancer and in development. Next slide. So this is the Wnt pathway. So you can see that beta-catenin is in the cytoplasm, and it can go to the membrane where it sticks to the cadherin, or it can go to the nucleus where it can interact with other transcription factors and regulate gene transcription. It can also be degraded, and its degradation is regulated by Wnts. So Wnts are growth factors or differentiation factors, if you will. And when they bind to their receptor, FZ, frizzled, they regulate phosphorylation of beta-catenin. So when beta-catenin gets phosphorylated, it gets degraded, ubiquitinated and degraded. But if it's not phosphorylated, it accumulates in the cytoplasm where it can go to the nucleus and affect differentiation. So very complicated functions for these molecules. Next slide. Next slide. So I've listed some of the functions of N-cadherin. As I already said, it's a cell adhesion molecule. It also regulates cell sorting, formation of junctions, cell polarity, growth factor signaling, apoptosis via the PI3 ACT pathway, chondrogenesis, stimulates mesenchymal stem cells to undergo chondrogenesis. This is very important to biomaterials, construction of biomaterials. And also it's a mechanosensor and acts the Hippo-YAP pathway. Next slide. So how does it mediate cell adhesion? It does so by a homophilic mechanism. What that means is a cadherin on the surface of one cell binds to an identical cadherin on the surface of another cell. And it does so via the trip residue at the N-terminus of the EC1 domain. And that trip residue inserts into a hydrophobic pocket. And the key residue in that hydrophobic pocket is the alanine residue that I mentioned previously. So that's shown in the structure's been worked out. And so you can see the tryptophan residue there in red sticking out of the EC1 domain, and it plugs into an identical cadherin on the other cell into the HAV site shown by the blue, the cyanin color. Next slide. So this is what I was showing when you start a company and the company gets a lot of investors and you turn out to be on the Toronto Stock Exchange. One thing you have to hire is artists. and you have to spend money on artists to get them to draw pictures so that you can show them to investors. And so investors can get a feel for what you're working on. And also you need them when you exhibit at a conference, like at an ASCO meeting or such, and you have a booth that costs you several $100,000, you need pictures like this to draw the crowds. So anyways, here's the artist's conception of two cadherins on opposite cells sticking together, causing adhesion. Next slide. So these are some things you never dream of spending money on when you're working in your lab late at night. Now, this is an electron microscope picture of an adherens junction. So what you see are two cells, and they're sticking together. And the purple is an adherens junction. So that's formed between two cells. And all those strands is a cytoskeleton, the microfilaments of the cytoskeleton. which plug into the adherens junction. The most important constituent of the adherens junction is N-cadherin, so that's what's mediating the attachment between the two cells. But it's not only a glue. So N-cadherin is not only a glue, it's a signaling molecule. So the adherens junction is actually a signaling platform composed of many different proteins. Next slide. Next slide, please. Thank you. Oh, one back. And there is what the signaling platform does. This is the interactome of N-cadherin, and it consists of 365 proteins, plus or minus a few, I'm sure. And what you see are four signaling what should I call them? Circles, I guess. Four signaling circles of interacting proteins. The primary one, number one, those are the proteins that bind directly to N-cadherin. And in the secondary circle are proteins that interact with the proteins in the first circle, etc. So these 365 proteins vary depending on the cell you have. So if you have a muscle cell, the composition of the interactome will be different than if you have an epithelial cell or if you have a cancer cell. So when you give an inhibitor of N-cadherin, you set off multiple signaling cascades, and those signaling cascades vary with cell type. So you can imagine the complexity of putting in an agonist or an antagonist into a tissue. Next slide. So I mentioned cell sorting. This again occurs during cancer progression. In carcinomas, for example, the epithelium first has E-cadherin, expresses a particular adhesion molecule called E-cadherin. And then when the cells progress and become cancerous, they switch over to N-cadherin. And one of the things that this does is it causes the cells to sort. So cells expressing E-cadherin will sort out from cells expressing N-cadherin. Next slide, please. So this happens during embryogenesis and during the cancer process. This is an experiment done decades ago, where you have two populations of cells, one labeled fluorescently, and that says, say, has N-cadherin, and the other cell population is unlabeled, so say it has E-cadherin. And you can mix the two cell types together and they will sort out, as you can see in A, B, and C with time. The other way to do the experiment is to let the cells aggregate and then put the two cell populations together, as in D, E, and F, and one cell type encircles the other cell type. So this is a very powerful way of causing cell populations to differentiate, to segregate. Next slide, please. Okay, and so this happens, as I said, the population initially has E-cadherin, some cells turn on N-cadherin, and the cells sort out. Next slide. There are a lot of junctions, different types of intracellular junctions. Epithelial cells have several, and they're shown here: tight, adherens, gap, desmosomes. None of these junctions form if you inhibit the cadherin function. So you can prevent the formation of all these different barriers just by inhibiting the cadherin. Next slide. And this just tells you what all the junctions do. Next slide. So you may have heard the epithelial mesenchymal or mesenchymal transition, where you have epithelial cells with many different kinds of junctions. And these cells mutate, some of them mutate and lose their E-cadherin. And this happens in cancer progression. So when they lose their E-cadherin, they lose all their junctions, and they turn on N-cadherin gene. And this N-cadherin is not as adhesive. It's phosphorylated in the cancer cells, and so the cells lose their polarity, become fibroblast-like, as I've drawn in the bottom caption, and become migratory and invasive. Next slide. So the driver for this EMT, epithelial mesenchymal transition, is TGF-beta, and cells. You can see this happening in various diseases, for example, where the epithelial layer gets injured. The cells, the immune system comes in, and the immune cells secrete TGF-beta, driving the cells into a mesenchymal cell state, and expressing N-cadherin. So N-cadherin is a mesenchymal marker, and E-cadherin, this different kind of cadherin, is an epithelial marker. And this is used, you'll see it all the time in journals when they're looking at fibroblasts and other cancerous cells. This is a determinant of a cancer cell type if it has N-cadherin. Next slide. One other important thing that N-cadherin does is it can interact with FGF receptor, and it does so in a side-to-side manner, as I've shown here in this diagram. So you can have complexes of N-cadherin monomer attached to a FGF receptor monomer floating around on the cell surface. Next slide, please. But when you get adhesion, N-cadherin mediated adhesion, that would bring the FGF receptor monomers into contact, and that would activate the kinase function of the FGF receptor, and then it would send signals to stimulate cell migration and promote cell survival. Next slide. Yeah. So this is say hello. Hello. My cat decided to join. I've lost my train of thought now. This just shows signaling cascades activated by FGF receptor. So if FGF receptor is activated, multiple different intracellular cascades are activated. The PI3 kinase act cascade, or the beta-catenin cascade, as I've talked about previously. So cell adhesion in the adherence junction platform is in itself a very powerful signaling platform, and then when you add to it the ability to activate FGF receptors, you even get more complexity in the response that a cell can have in response to adhesion. Next slide, please. Okay, let's talk about antagonists. As I showed you before, antagonists are based on the tryptophan N-terminal domain in red, or the HAV subdomain in cyanin. So the antagonists are based on those two subdomains. Next slide. Thank you for the comment about my cat. So as I said, N-cadherin promotes cell adhesion, and what the inhibitors do is they're small molecules and they come in and they bind to the hydrophobic pocket and prevent the N-cadherin monomers from interacting. So it's a simple lock and key and mechanism. Next slide. So this is the development of antagonists over the years. The first antagonists were linear peptides, which were discovered in 1990. And then they weren't very useful because the half-life of a linear peptide in the bloodstream is just a few minutes. So if you inject the linear peptide into the blood intravenously, it degrades almost instantly. So you have to do other tricks to increase the half-life. And one trick is to simply cyclize the peptide. And so that is the foundation of ADH1. ADH1 is simply a cyclic peptide with the HAV sequence. Many years later, and ADH1 was the basis of Adherex, my first company, which went public. After cyclic peptides, we went on to make small molecules like LCRF and CRS. Peptides can't be taken orally, whereas small molecules can be taken orally, and so it makes them much better to administer, much easier to administer. Also, small molecules don't have peptide bonds, so they are not rapidly degraded and last for a longer period of time in the bloodstream. So you do want to progress to making small molecules. They're also easy to manufacture and quite cheap, so they make ideal drugs, as opposed to, say, the current trend, which is monoclonal antibodies or CAR T cells that therapies that cost $200,000 US a treatment. These small molecules cost barely anything as drugs. To my mind, small molecules make the best drugs, is I guess what I'm saying in my long-winded way. The small molecule CRS is 1,000 times more potent than ADH1, the cyclic peptide. So this has been a tremendous improvement over the original antagonist. And these molecules, this class of molecule is the basis for my new company, Zonula. Next slide. So it's important that despite knowing that N-cadherin is an adhesion molecule and has many functions, it's important to note that N-cadherin antagonists are not toxic. ADH1 has been in human beings, has been used in human beings, and the maximum tolerated dose was not found. Also, N-cadherin monoclonal antibodies are not toxic in mice. They haven't been used in humans. And the small molecules have so far shown no toxicity in animals. The CRS, the most potent one to date, as I said, it's easy to manufacture, it's not toxic, and it's orally available. So it has good properties to be a drug. Next slide. So as I said, these antagonists disrupt cell adhesion. This just shows ADH1. It's a very old slide. What you see is a monolayer of cells at time 0, and the cells have N-cadherin on the cell surface. That's the green line. The blue is the nuclei of the cells. And if you add it to cells in culture, a monolayer, you can see with time the staining, the continuous staining starts to disappear. Next slide. And within a few hours, cells start to separate from each other, and you can see gaps between the cells in the monolayer. And then after about 6 hours, the cells start to retract from each other, and you can see that the morphology has changed from cuboidal to a more fibroblast-like morphology. Next slide. And finally, after 12 hours, the cells have entered apoptosis, and the staining has disappeared. So this is the kind of thing you see with either an antibody against N-cadherin or a N-cadherin antagonist in vitro if you add it to cell cultures. Next slide. So what happens when you add an antagonist to endothelial cells, though, is the effect is much more rapid. So endothelial cells are much more sensitive, and within 1 hour, as you can see, already the cells have rounded up, and after 6 hours, the monolayer is totally destroyed. So, as I said previously, the effects of the antagonists vary depending on cell type and the conditions of the culture system. Next slide. Next slide, please. All right. So go back one. Yeah. So go back. Perfect. Don't move. So we're investigating N-cadherin antagonists for use to treat fibrotic diseases. So fibrosis is quite simply the excess deposition of collagen, and the collagen is disorganized due to disease process. It's heavily deposited and causes scars to form. The main cell producing the collagen is a fibroblast, and the fibroblasts express N-cadherin, as luck would have it. And we've shown, and others have shown, that N-cadherin antagonists can block not only fibroblast adhesion, but over time kill those cells. They could also affect collagen synthesis and deposition by the fibroblasts. And so they theoretically can be used to treat fibrotic diseases. Next slide. So we're trying to use our antagonists in several programs. We're investigating their effects on pancreatic cancer. Many people don't realize that cancers can be considered fibrotic diseases. For example, pancreatic tumors can have up to 90% fibroblasts. So they can be considered, this type of cancer can be considered a fibrotic disease. We're also investigating the effects of our antagonists on scar formation and also on pulmonary diseases such as idiopathic pulmonary fibrosis. The programs are not very developed. We're trying to get more money, obviously, to develop these programs. Next slide. Okay. So in 2020, a paper appeared in Nature where they used ADH1. It wasn't us, it was another group. They used ADH1 and they showed that it could prevent scar formation. And what they found was that in the skin, there's a progenitor population of fibroblasts. And when skin is damaged, cells of the immune system come in to repair the wound, and they secrete TGF beta. And this TGF beta stimulates the fibroblast precursor population to divide and multiply and differentiate. And what these cells do is they swarm to the wound. They aggregate and they swarm into the wound, and then they lay down collagen and close the wound. But the collagen formation is so uncontrolled that you get a scar. So if the swarming and the aggregation of fibroblasts turns out to be controlled by cadherin. And so what they found was that when they added ADH1, the cadherin antagonist, it prevented the fibroblast adhesion, the aggregation, and the scar formation. Wild. So based on their observations, we're looking at the effects of our new compound, our new antagonists on fibroblast aggregation and wound healing. Next slide. So with regard to idiopathic pulmonary fibrosis, this is much the same story. The lung epithelium gets damaged, for example, by smoking, by inhaling pollutants, what have you. And the damage causes the immune system to become activated and immune cells to go to the site of the damage. Again, they secrete TGF beta, which stimulates the fibroblasts to proliferate and heal the wound. So we think that cadherin antagonists would be useful for treating a variety of pulmonary diseases. Next slide. And finally, the most advanced program that we have going is on pancreatic cancer. As I mentioned, the pancreatic cancer tumors are composed mostly of fibroblasts. And consequently, the cancer cells are hidden from the immune system and from drugs. Drugs are prevented from getting to the cancer cells by the fibroblasts and by the collagen that they secrete. So we and others have shown that cadherin antagonists block collagen deposition in pancreatic tumors grown in mice. And so we think that these antagonists can be used to treat pancreatic cancer. Next slide, please. So I just want to emphasize that many cell types express cadherin. So in a tumor, you have cancer-associated fibroblasts. They have cadherin. You have the cells of the blood vessels, endothelial cells and pericytes. They have cadherin. If there are metastatic cancer cells, they are likely to have cadherin. Obviously, the cancer cells themselves have cadherin, and the stem cells have cadherin. So when you give the antagonist, you're not only attacking the cancer cell itself, but you're attacking multiple cell types within the tumor. So this is a really a different way of treating a tumor. We're not just attacking one cell type, we're attacking multiple cell types. Now I've put this inset in this table at the bottom, showing the relationship between the parasites and the endothelium of blood vessels. The parasites are smooth muscle cells that contract and give rigidity to the blood vessel. So blood flow through the blood vessel is regulated by the parasite. And these cells are often ignored. I must say it really bothers me because I review many papers every year for various journals, and people culture endothelial cells, and people do endothelial cell tube formation assays, and they say, well, we're looking at angiogenesis, and we're looking at blood flow through the tubes. Well, this is not an in vivo situation. You need the parasite to actually figure out what effect your drug is having on the blood vessels. And as you can see, the relationship between the parasite and the endothelium is very different. If you have a cancer cell, like the one on the right, the parasites are barely sticking to the endothelium. Whereas if you have a normal capillary, shown on the left, the parasite is like an octopus, and it sends out its tentacles and wraps around the endothelium. So the relationship is very different, and a lot of it is due to the VEGF that's secreted. VEGF binds to VEGF receptor, which is a tyrosine kinase that phosphorylates everything and makes adhesion weak. So this kind of in vivo, in vitro discordance is very troubling to me, especially when you're looking at the effects of cancer drugs in vitro as opposed to in vivo with various model systems. Okay, next slide. And this is just a next slide. I don't need to talk about this. Next slide. Next slide. I've hammered. Yeah, good. Stop. So let me just show you some examples of cadherin antagonists and their effects on tumors. So this was published a number of years ago, not by my group, but by another group, showing the effect of ADH1 on pancreatic tumor growth in mice. Next slide. And so what they did was they grew the cancer cells in mice, and they were labeled so you could monitor the tumor growth. And then they injected the mice with ADH1, and what you can see is the antagonist slowed tumor growth dramatically. But you'll note it didn't stop the tumor growth. So this emphasizes the need for combining drugs to treat tumors, and I think that's what everybody's finding is you need to combine various tumor drugs together to get maximum effects on tumor growth. Next slide. Next slide. Yeah, so this at the bottom is a very dramatic, this is a melanoma tumor growing in a mouse, and on the right is a mouse that's been treated with ADH1 and another cancer drug called melphalan, and you can see it is very effective in treating the melanoma. Next slide. There's we stumbled across, how should I put it? we found that with Stephanie Willerth, who you might know, I'm sure you know her. We did a project where we looked at the effect of my latest small molecule antagonist, CRS, on glioblastoma, and we used her organoid cultures to look at the effects of the drug. And what we found was that our drug killed the glioblastoma cells. So we published that, and then I started looking in the literature more carefully, and I found that another group in Belgium many years ago had shown that ADH1 killed neuroblastoma cells in vitro. And then, surprisingly, a paper just came out showing that ADH1 killed meningioma cells in vitro. So all of a sudden, in a span of a few years, N-cadherin has emerged as a therapeutic target for brain cancer, 3 different types of brain cancer. So this is really worth pursuing. The problem is these cancers are very rare, and so you need to interest a neurosurgeon or a group of neurosurgeons to get your drug tested. So we haven't made much progress in developing N-cadherin antagonists for brain cancer, but I'm putting it out there and I'm publicizing it as much as I can to hopefully get a neurosurgery group interested in working with me to develop these drugs for brain cancer. Next slide. And I just want to show you 2 cases of human beings with cancer that were treated with ADH1. Next slide. So this was a woman with a carcinoma that spread to the lungs, and she was treated over a period of months, and they did CT scans on her lungs. So I don't know if you can see this red circles, but those are metastases in the lungs, and as you can see with time, they're reduced and disappear. So this was a very dramatic indication that N-cadherin antagonists can have an effect on tumor growth in human beings. Next slide. And this is just a second case I want to show you. It's an adenocarcinoma of the colon. Next slide. And in this case, the tumor had been treated with other drugs, which show with the green arrows, and the tumor kept growing. So the known treatments weren't affecting this tumor. But the red arrows are ADH1 being administered at various time points. And as you can see, the tumor stabilized. It didn't grow more, but it also didn't decrease. So the drug did have ADH1 did have an effect, whereas other drugs didn't. Again, encouraging us to think that N-cadherin antagonists can be effective anti-cancer drugs. Next slide. And I think I'm done. Oh, no, I'm not done. Sorry, agonists. Sorry, I completely forgot. So the first, this will be quick, because it's new. It's a new area. The first agonist was the cyclic peptide, which had 2 HAV sequences, HAV and HAV, dimerized. That's on the left. And the way it worked, it is thought that it coupled N-cadherin monomers together, causing intracellular signals to be sent into the cell. And this antagonist was very good in stimulating neurite outgrowth. So the thought was it could be used in neural regeneration applications. Biomaterial-wise, this field has gone crazy. They've been attaching linear peptides, HAV-containing peptides, to various supports. This particular one is a self-assembling polymer, and the sequence is at the top: KLD, whatever, da 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 da. So if you embed mesenchymal stem cells into this polymer, turns out they'll differentiate. Their cadherin will be turned on, the signaling pathway will be turned on, and they'll differentiate into chondrocytes. Next slide. So the field, everybody's making HAV biomaterials. So this one, the HAV sequence is shown there, HAV, DI, GG, what have you, and it's coupled via cysteine, so a disulfide bond, in other words, to the support, to the hydrogel. And this hydrogel was actually injected into the knees of rabbits, and it stimulated chondrogenesis and cartilage formation. Wow. So this is a very active field, and I would say that this is not the best peptide to use. As I've said, I have new compounds now which are 1,000 times more potent than this compound. So we'll see how this all works out in the end, but obviously I'm interested in making biomaterials now, agonists instead of antagonists. Next slide. This is just another biomaterial that's been made. Next slide. Okay, so just to end then, there are a number of antagonists and agonists that I've talked about. They're in various stages of development. Some are linear peptides, some are small molecules with different backbones. So for example, imidazole or pepridin. And we believe they have a large number of therapeutic applications, and I've just listed some of them here. And I'll end with that. Thank you for listening. Oh, my collaborators. This is a number of people through the decades, actually. But I've highlighted some significant contributors, especially to this talk. I particularly point out Arkady Weisberg and Axel Thompson. Arkady is a superb chemist who designed the small molecules, and Axel Thompson is an expert in fibrosis. So they've been intimately involved with me driving the work. I've also mentioned Spiro Getzius, who some of you might know. He introduced me to Stephanie, so he was a former student of mine, as was Yannick Pouliot, who worked with me on the initial linear HAV peptides. Thank you. Okay.
Well, thank you very much, Dr. Blaschuk, for your very interesting presentation. I really enjoyed it, and then I have a couple of questions to ask from you. I'm not sure how much time do you have that after your talk. We typically stay for like until for half an hour, but it's we know that you have a busy schedule. So if at any time you feel that you need to leave the meeting, just please let us know, and then we can stop the Q&A. So we will start with.
I'll only answer easy questions.
All of them will be very easy, actually. So let me start with the with a question from the audience, Alex. How would you limit the area of apoptosis? I'm not sure if, in the context of probably using and cadherin antagonists, probably the application for apoptosis.
How would I limit it? Well, to begin with, if it doesn't occur instantly. Apoptosis of the cells doesn't. So if you give the compound and you to the cells, let's say in a culture dish in vitro, and you wait 2 or 3 hours, and then you remove the media and put fresh media on without the antagonist, the cells will come back to life. They won't die. So it's reversible to begin with. If you leave it a long time, then the cells will die. So in the body, if you inject the compound into the body of an animal, the animal doesn't die. This puzzled me tremendously. I thought that the animal would melt, actually. But it didn't melt, and it was fine. And I guess the dose was not high enough, and it was cleared from circulation too quickly to have an effect. So if I gave the drug continuously at a high dose for a long period of time, I probably would see the animal dying. But it's not given that way. So does that kind of answer your question? I struggle with this all the time. There are a lot of different adhesion molecules at play also, I should add. So just because you inhibit one system doesn't mean you trigger it. It's all cell context dependent is what I'm trying to say. So if you take an endothelial cell by itself, it reacts differently than if you take an endothelial cell and a pericyte that are together in a blood vessel structure. And indeed, what they found is if you knock out N-cadherin, the blood vessels get leaky, but the animal doesn't die. So you can have an animal where N-cadherin is dysfunctional. And that's because the integrins, there are other systems like the integrins kick in and stabilize the blood vessels, but it does become leakier. Yeah.
So
you can make cells do anything you want them to in a dish. Okay. And so that's, as I'm sure all you bioengineers know, you can make cells do anything in it.
Well, now that's there are many questions now that I have to ask as a follow-up to this a question, and then also your answer is that so my first question is about the route of administration of let's say cadherin antagonists. So let's say in the case of cancer or fibrotic diseases. So what is the envisioned route of administration? Is it going to be orally administered or IV or how?
Exactly. So the low hanging fruit is scar tissue formation and applying it topically. So right. So if you have a scratch or a cut, you would apply topically. So it wouldn't bother the whole body. It would just be a localized. Put it on a Band-Aid and slap the Band-Aid on the scar. In the case of brain cancer, the surgeon. The first thing that happens is the surgeon goes in and takes out the tumor growth, and then administers often in a wafer, a poison. So you know that the most used cancer drugs are DNA inhibitors,
Yeah.
the DNA synthesis. They're totally non-specific, but yet they're the most effective drugs available. So he would put a wafer on with the drug onto the area where the tumor was in the brain. So again, that would be a localized, crude but localized delivery. In the case of fibrosis, I would use a nebulizer. So, I guess what I'm saying is injecting the compound would be my least favorite route of administration.
So more, yeah, what you're thinking is a more localized therapy approach.
Yes. Yeah. Now, ADH1 was administered intravenously because it was a cyclic peptide, and the only way you could administer it was intravenously. They administered it over a period of 1 hour, and then rested the patient. As you saw in that one, they gave it once every 3 weeks. So it had an effect, even though it wasn't administered continuously.
Because I was worried about, you talked about making the blood vessels leaky, and then I was worried about the issue of internal bleeding, and then some other issues.
Absolutely. And as I said, if you were going to administer it continuously, that would be a real worry.
Yeah.
Injection. Well, I mean, we have injected it, and we look at the injection site, and it looks okay. So I would expect that if it had an impact at the injection site, it would turn all red and get bloody. But we didn't see that.
But in terms of long-term administration, then because these therapies, I mean, typically these chemotherapies, you need to administer them for like periodically, but in the long term to see some sort of, at least the chemotherapies that we see. But this is something that can be definitely studied, and that could be a very interesting project. But I have
slow releasing gels. Yeah, absolutely.
So, so you talked about the application of N-cadherin antagonists for various types of diseases, and one type of disease or organ caught my attention, and that was pulmonary fibrosis. And then I was reading that patients who who had COVID, they will like this fibrotic tissue is formed in their lungs after the diseases. So I was wondering if there is any benefit in using these types of drugs for post-COVID treatment of patients who had this disease, or in general for infectious diseases.
Well, if you get any kind of scarring, any kind of scar formation, you could envisage using an N-cadherin antagonist to prevent scar formation. I was reading, like, for example, spinal cord injury. One of the problems repairing spinal cords is that scars form. You get collagen deposition, and you have scars, and they prevent the neurites from growing back. So this phenomenon about cell aggregation and swarming has been seen in tumor cell migration, for example. In neuroblastoma, they migrate as a cell collective, as it turns out, not as a single cell. So this idea that metastasis involves single cells moving, that's wrong.
Mhm.
that's wrong.
Mhm.
The cells seem to move as a collective, just like with the scar tissue that I showed you, uh, where the fibroblasts in the skin swarmed and worked as a collective to put down the collagen. Yeah. So
Yeah, yeah, yeah.
yeah, there's a lot to be figured out. That's for sure. And I'm continuously surprised at at uh, at all the applications that emerge. When I started 35 years ago with ADH1 and thought, oh, it was just going to be used for cancer, and now I find it can be used for maybe headaches.
Mm.
You know, it's almost like aspirin. Uh,
Very interesting. Very interesting.
because the stroke is a decrease in blood flow to the brain, right? And
Yeah.
what happens, they just published, what happens is it's the capillaries in the brain, the pericytes freeze up and clamp the blood vessels shut. So if you have something that can relax the parasite, as soon as the guy has a stroke, you want to give it and get that
Mhm.
blood flow going as fast as possible.
Mhm.
So this is like, I don't know where this application came from, but there it is all of a sudden.
Mhm.
Because
Agreed.
people didn't appreciate parasites. That's why I was going on and on about it. Um, I was introduced to the concept of a parasite by Elisabetta Diana from Milano, who introduced me to risotto also, and she never got around to telling me about the difference between men and women, but she was
Mhm.
going to at one point. She said, "I'll tell you in the future." But she introduced me to parasite, and she was a superb vascular biologist, and she knew all about them.
Mhm.
But 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 nobody was even talking about them in the cancer field. Nobody was talking about them in the brain field, and now it's like they're the cell to concentrate on when you're talking about vasculature.
Mhm.
It's
Yeah, I agree.
and,
I agree.
yeah, yeah. And
So,
again, and also the fibroblast. All of a sudden, everybody's talking about fibroblast and tumor microenvironment, whereas a few decades ago, no, it was just the cancer cell. It was all about the cancer,
that's
right?
because probably bioengineers they started developing these 3D models, and then they were started thinking about mimicking the actual or the native tumor microenvironment in which you see all these different kinds of, you know, components of the tumor microenvironment, including the parasites, and then also the vascular system, and then the stroma cells in general, and then also the immune cells. I just wanted to there's one comment from JP May from participants. It's about collaborating with neurosurgeons, and then they were suggesting that as an alternative to collaborate with neurosurgeons, you may want to collaborate with a team working on ultrasound-based drug delivery systems. That we actually, I mean, in LA, where I am right now, there's at UCLA, there's a neurosurgeon who is actually developing this ultrasound-based system that basically makes the blood vessels leaky, and then it allows for the cells to penetrate. And then that could be one application as well for localized therapy. Maybe using and cadherin antagonists. You said that it makes the blood vessels leaky, right? And then if you can locally make the blood
I
vessels
would,
leaky, then you can deliver the drugs that do not cross the blood-brain barrier, and then that could be
I
used in
would love,
combination.
yeah, I
So,
would love to see the effect of the antagonists on blood flow a normal blood vessel blood flow.
So there
I'd
are.
love to do those experiments.
So I know a few people actually at Toronto, and then also at Sherbrooke, which is very close to New York, who have animal models of glioblastoma and then other types of brain tumors, and then they basically their focus is on the blood-brain barriers, and I can introduce you to them. So you guys can define a nice project, because that could, I see there is some potential in developing drugs to the brain by using this cadherin antagonist. So my next question is about the expression of cadherin in materials, in cells, and whether you have studied on the different levels of expression of these molecules in resistant cells and non-resistant cells. So we have one of the challenges with cancer, as you know, is chemoresistant. And then I wonder if there is any study that shows the expression of these resistant cells and cadherin in resistant cells versus non-resistant cells.
Well, that's why I was talking about fibroblasts the compounds don't affect the cell. If it doesn't have cadherin, we don't see any effect.
Mm.
And also, if the cells have so there are some cells that have cadherin, but the compound doesn't affect them.
Mm-hmm.
So, for example, when we did the work with Stephanie, we co-cultured glioblastoma cells with astrocytes in organoids, and the astrocytes weren't affected, even though they have cadherin.
Mm-hmm.
And so they obviously have different signaling pathways in play. You can't just say this cell has cadherin and therefore it's going to respond this way. That's why I showed the interactome. That interactome was for actually cardiac muscle cells. It wasn't for, so if I did an interactome for a glioblastoma cell, I don't know what I'd find. Well, I know the basis of what I find, but I don't know what signaling pathways would be affected.
Mm-hmm. Mm-hmm.
And it all has to do with the phosphorylation level, I think, of the cadherin and the catenins. I didn't talk. I didn't talk about the tyrosine phosphatases and tyrosine kinases that affect cell adhesion.
Mm-hmm.
But there's a whole repertoire of those in different kinds of cancers.
Mm-hmm.
We found the oncologists found that gynecological tumors, there was an indication that they were being affected quite a lot with ADH1, that particular type of tumor, ovarian cancer, in other words. But what happened is we just ran out of money. It was 2008. I don't know if you guys know about 2008, but that's when the stock market.
Yeah.
And in Canada, biotech was wiped out. It was unbelievable. It was wiped out. We were one of the largest biotechs back then. We had 50 employees, and the money just went away. It was unbelievable. These people, they have billions of dollars to invest, the big VCs, and if they lose 20 million, it's nothing to them. It's hard to believe, I know, but it's nothing to them. They just walk away.
Mm-hmm. Yeah.
So that's what could happen right now, this year, with the war raging. Yeah. Anyway,
Yeah,
another
yeah,
topic.
yeah.
Money is a very big topic when it comes to doing research, and I had a bunch of slides that I actually threw out that I was going to show on the cost of doing development of one drug. One
Well,
cancer drug.
that's what I wanted to ask you actually as a next question. Maybe briefly.
Yeah, $1 billion for a cancer drug to get it into a human being. So that it's I don't know what the budget of CIHR is, but I bet you it's not $1 billion.
A couple of $100 million, but I don't think you can.
The amount of money that drug companies spend and come up with nothing. I had a slide that I threw out of this talk, but there was a time when GSK was spending 3 billion pounds a year on drug development. 3 billion, and for 10 years they had no drug. No drug came out of that company. And you can look now, right now, today, there are many big pharmas that have nothing. Even though they're spending billions, they have one drug that's a blockbuster, and when it loses its patent, they got nothing to follow it up. It's unbelievable to me. I can't explain it. Because if you gave me $1 billion, I for sure would come up with a drug, as I'm sure you would too. You'd come up with something.
So I'll just ask one last question as a follow-up question. What do you think is the challenge? I mean, you brought up a challenge, right? What do you think is or you brought up a problem? So there's like billions of dollars being spent on drug development over the past few decades, but as you mentioned, some companies, they didn't even develop one single drug that could be useful. How do we solve this problem? What are the solutions, or what do you think an ideal solution would look like?
Well, we can cure cancer in animals, right? Right?
We can.
We can cure cancer in animals.
Yeah, definitely.
Because our model systems aren't good, and I think that's what you guys are all about, right? You're trying to develop appropriate model systems for testing the drugs that would help. I mean, that's the thing. You start with 1,000, and that's what they do. They start with thousands of candidates for a particular target, and they narrow it down to 5, and then they spend millions on those 5, animal testing, this, that, get one out of that, and then spend $1 billion to get FDA approval and turn it into a drug. And maybe it'll work and maybe it won't. It's the whole process is unbelievable. Mm-hmm. So now they're going with personalized medicine, and they think that might be the solution, is to very carefully choose your group of treatment candidates, your group of people, and see what their genetic makeup is like, and characterize every person as carefully as they can before they start a clinical trial. And then they will have success. Mm-hmm. And I'm sure, I mean, that's why we can cure mice. It's because we take one strain of mouse and we genetically engineer it, and then we put in one cell line into that mouse and get a result. But that's not what happens in a human being. So if you have like a KRAS mouse that develops tumors spontaneously, you mutate to overexpress RAS, say, in pancreatic cancer. That's a model. The tumors develop on their own pace. So at any given time, one mouse will have a big tumor and one mouse has a small tumor, but with time they all develop tumors. You more approximate what happens to a human being, why my tumor grows faster than your tumor, and why my tumor responds to the drug and your tumor doesn't respond to a drug. It's all about characterizing the genetics and narrowing down the treatment group, which is what they're doing now. They're sequencing like crazy every person with a particular disease, and then throwing all this information into a computer and letting AI kick out the candidate populations for a particular treatment. That's what it's evolving to now. Yeah.
Well, great. Thank you very much, Dr. Blaschuk. I mean, actually, it was, very interesting talk with us. This is so we typically have speakers in this area who talk about these disease models and then how the drugs can be used. But you covered the area where the drug is being developed, like the basics of drug development based on a specific molecule that is expressed in a certain disease. So that's a very nice addition to what we had so far. So Hooman, do you have any comments or questions?
No, I was amazed by the presentation and I really enjoyed and learned a lot. So, this is really something that we need to bring it to the market, especially these kind of medications or drugs. And in terms of parasites, also, we have colleagues here in San Justin Hospital, because you have been at McGill for a long time, that they're working on parasite retinotropy and other things. So they have animal models and stuff. So if you want, I can introduce also.
Email me suggestions. The technology, that's why I started with cell adhesion technology. It can be applied to many things, and I'm only one person, so I need to collaborate. I'm all about collaborating. That's.
And Hooman is also
exactly. So I pass the mic. I thank you so much.
So I just wanted to add that Hooman is also working on cardiac diseases and cardiac tissues, and then here's one of the areas. Yeah.
So we are looking now. Exactly, we are looking now with this colleague who study parasites and this leaky lumen on endothelial layer. We are trying to do printing and this kind of bioprinting stuff and look at the models and the stuff. So this is also a model that we're looking at.
If you have a biomaterial and you put on the cell, the endothelial cell, and you have an HAV biomaterial, that endothelial cell will think it's sticking to a parasite. Okay. Yeah.
Exactly.
So yeah.
Thank you.
Awesome.
This was amazing. I learned a lot. Thank you so much.
And then again, I'd like to also thank the participants for joining this very interesting talk. I'd like to again reiterate my appreciation to our speaker who talked about a very interesting topic today. So with that, I'd like to conclude this session and then see everyone in the next talk. Take care.
Bye.
Take care.
Stay safe.
Bye-bye.